Hello guys, welcome to Big Book. Today we are going to discuss a new topic, that is the dosage compensation of genes. It can also be understood by Lyon's hypothesis, Barrbody, X chromosome inactivation, chromatin body, sex chromatin, and etc. Dosage compensation of genes. This is considered to be as the hypothesis earlier. Later it is considered to be as the phenomena, and a hypothesis is explaining that the sex chromatin body and dosage compensation is in mammals is determined by the inactivation of one X chromosome in the normal female, and the hypothesis was named after Mary Lyon. It is also called as Mary Lyon's hypothesis. Lyon's hypothesis demonstrated that to the number of the X chromosomes is active, that is one active X chromosomes and rest are the Barr bodies. Here for example, XX female and in XXY male one Barr body is found and the one active X chromosome is found, whereas in XXXY males and XXX meta females it is two Barr bodies seen, that is one X chromosome is active. So here the formula goes like this: n of x minus 1. N stands for the number of the sex chromosomes present, that is the number of the X chromosomes are present. So here in the normal female, that is the XX, the sex determination is XX, so 2 X chromosomes are present, so N will get the value of 2. So 2x minus of the 1x is equal to the 1x remaining. So 1x minus of 1x nothing but, in the formula minus of 1x is nothing but the 1x is going to remain active out of the whatever the number of the sex chromosomes are present. So here it is understood that whatever the total number of the chromosomes will be present, that is the total number of the sex chromosomes are present or the X chromosomes are present, those X chromosomes to the total number of the X chromosomes, one X chromosome remains active and the rest remains the inactive X chromosomes in the female sex. So here in the normal, if you are going to consider the XXY male sex, in this also the dosage composition is going to happen, that is out of the 2 X chromosomes, one X chromosome is going to remain active and the other X chromosome is going to become inactive. In the same manner, if you are going to consider XXXY, it is also a male. Here there will be one X chromosome active, the other two will become inactive X chromosomes. And if you are going to consider XXX females, those are also called as the meta females. In this also there will be 2 X chromosomes which are going to form the inactive X chromosomes and one is going to be the active X chromosome. So the whatever the number of the inactive X chromosomes are present in the cells, those are called to be as the Barr bodies. So next slide. Here in this slide you can see the different examples. Even in this we have mentioned normal male and normal female. Normal female will be having the one Barr body, normal male will not have any Barr body. If you are going to consider, this will be the normal female and this will be the normal male. And if you are going to consider here, this is the syndrome trisomy, that is XXX is the trisomy syndrome, which is going to have the 2 Barr bodies, but the total number of the sex chromosomes are 3. Now here if you are going to see the Turner syndrome, the total number of the sex chromosomes are 1x, and there will be the absence of the other sex chromosome. So here it is going to have 0 number of the Barrbodies. And the below Klinefelter syndrome, here you can see XXY as the sex chromosomes. So here you can see 1 number of the Barrbody. So here this is a small table which is giving the exact meaning what Mary Lyons is explaining. As per the Lyon's hypothesis, the number of the sex chromatin bodies in the interphase cells of adult females is 1 less than the number of the X chromosomes observed in metaphase preparations. Now here it is a very brief point. As per the hypothesis, we understood that one X chromosome remains to be as the inactive and the other will be active. This is what we are going to observe in the interphase or the interphase form of the cells, where one chromosome is going to remain as the inactive, that is, it is going to not appear as the normal sex chromosome or the X chromosome. But whereas in the metaphase, both the X chromosomes are going to appear in the form of the sister chromatids. So here the explanation is like that. After the interphase, whatever this cell is not showing one of the X chromosome has to go through the somatic cell division or the mitotic cell division, that is the equational cell division. So here after the interphase, definitely the sex chromosomes, whatever the Barrbody or the inactivated sex chromosome will be present, it is going to get duplicated. And the synthesis of this particular chromatin matter is going to take place. So later in the metaphase, where both the X chromosomes are going to be seen, and that is going to equally divided again, but with the next daughter cells whichever is going to happen, that daughter cells again are going to show the interphase to be having one X chromosome to be inactive, that is less compared to the total number of the X chromosomes or the sex chromosomes should be seen. Only one X chromosome is required for normal metabolism in females. The additional X chromosome are condensed and becomes heteropyknotic. Now here, when you are going to consider in the normal females, there will be 2 X chromosomes. Out of this, one will be active and other will be the inactive. Hence the one is sufficient for the metabolism and the other is going to prove the heteropyknotic. Heteropyknotic is referring towards the chromosomes which are densely or highly condensed. So here this densely or highly condensed form of the chromosomes are going to take the high stains at their locations. So here, as chromosome is going to become heteropyknotic, it is going to be heterochromatic in nature. So by referring towards the name of or the concept of taking high stain regions, it is considered to be the chromosome is going to form the highly condensed X chromosome and it becomes heteropyknotic when it is stained. Hence mammalian X chromosomes are of capability being heterochromatic in some cells and euchromatic in others. So here it is mentioned to be as the sum ratio of the difference which is observed in the studies. Here in the labs also students will come across that in many studies it is observed to be as the 50 to 70% of the cells It is the range of the ratio where if you are going to consider a normal human female cell studies for the Barrbody or the sex chromatin, inactivated sex chromatin, in this if you are going to consider out of from 50 to 70% of the cells, whatever the sample will be having, out of the 100% of the cells, out of 50 to 70% of the cells will show the Barrbody, the rest will not show the Barrbody. So it is understood with that in certain cells the X chromosomes are active, they are euchromatic in form, but in some cells they are going to remain the Barrbody or the heterochromatic. Lyon's hypothesis has certain genetic consequences of genes in mammals. Dosage compensation in females has led the single X chromosome to regulate enzyme activity to the level of males, with only one X chromosome. So here dosage compensation is nothing but the number of the expression of the genes, that is the genes which are present in the females. That is, if you are going to consider in females, on the X chromosomes, on the both the X chromosomes there will be the homologous form of the genes, the locus will be having the similar genes, but because of the inactivity of the one of the X chromosome, the one fold of the X chromosome gene expression has been suppressed or lessened. So here, because of that, the dosage compensation has been formed. Whatever the X chromosome is expressed in the females, the same amount of the fold or the fold of the gene expressions has been regulated in the males also. In the random and large sample studies, it is observed that there is a variability in expression of heterozygous females because of the random inactivation either of the two of the chromosomes. Evidences supporting hypothesis of dosage compensation in mammals. Females occur through inactivation of all but one X chromosome, thus producing functional equivalence in the two sexes. So this second point, in this it is said to be that, for example, in a female there will be two X chromosomes where both the X chromosomes has been inherited by the two different parents, where both the X chromosomes will be having the different number of the genes, which is going to have different kind of the triad combinations in the form of the alleles. So this, because of the one X chromosome putting on to be as on the inactivation process, the other is going to express. So the inactivation process is so random that it cannot be expected that this chromosome with this allele is going to become inactive in the next generation. So out of the two, any one X chromosome can get inactivated. So it is such a random process that the variability in expression of heterozygous females because of the random inactivation has been seen. It is out of the either of the chromosomes. So evidences already we have spoken about already, that the functional equivalence in the two sexes because of the gene expression. So in studies of glucose 6-phosphate dehydrogenase activity in cells, the enzymatic activity was shown to be about equal in two sexes. That is, activity in females with two chromosomes was approximately equal to that in males with one chromosome. So here the studies, that is during these studies, that is to understand that one is that the one X chromosome is going to be inactive. So during that time they have made a study in the females cells where they have taken the skin cells of the human females to study the glucose 6-phosphate dehydrogenase activity in the cells and the enzymatic activity, and they have made these particular cells to be cloned. So that is, activity in females with 2 chromosomes was approximately equal to that in the males with 1 chromosome. Glucose 6-phosphate locus has 2 alleles. Electrophoretically produces distinct enzymes F and enzyme S, and are tested with heterozygous females. So here already I have spoken that glucose 6-phosphate dehydrogenase is a gene which is having the 2 distinct kind of the alleles, which is going to form the enzyme F and also the enzyme S because of the heterozygotic form. So here, because of this, here they are studying is the chromosome in the sample, in such a sample where the X chromosomes, both the X chromosomes are heterozygotic for the locus of the glucose 6-phosphate, that is F and S. So they are taking the sample and they are studying for the same, and they are going to clone. The biopsy of the skin from these heterozygous females were cloned. Each clone contains either the F or the S form of the enzymes. So here, to study the random inactivation, they have taken this sample. So here, when they have cloned the sample, whatever the cloned sample of the F has given the F kind of the culture itself, but not the other form. Whatever the S kind of the culture, cloned culture, has given the S kind of the results itself. So this support that one X chromosome received from either the mother or the father of the female was inactive. So here, out of this, the whatever this random number of the random form of the X chromosomes has been received from the parents, out of that one remained inactive, and it is going to remain inactive for the whole life cycle of that particular organism. As the inactivation has occurred, the clone of cells derived from one progenitor cell all express the same allele. In human embryo, X chromosome inactivation starts in the late blastocyst, about the 16th day of life. Once the inactivation is established, it is irreversibly maintained in somatic cells. In germ cell line, reactivation occurs at a specific stage of germ cell development. It is in meiotic prophase. So here it is clearly explained that in the early blastocyst, when the cells are starting to divide, with the late blastocyst, the formation of the Barrbody will takes place. So in germ cell line, reactivation occurs at a specific stage of germ cell development. It is in meiotic prophase. So during germ cell line, the reactivation of this particular inactivated X chromosome will takes place to take part in the segregational division of the genetic material. So Barrbody name has been given against Mr. M.L. Barr, who has found this in the female cats, the X chromosome, which is going to remain as the densely stained particle on the nuclear membrane. So the name has been given to be as the Barrbody. Is a small densely staining structure in the cell nuclei of females consisting of a condensed inactive X chromosome. Mammalian chromosome inactivation is initiated from the X inactivation center or XIC. So here in the X chromosomes there will be a locus that is called to be as the X inactivation center or XIC. So the inactivation center contains around 12 genes. Out of this, 7 will be coding for the proteins and 5 will be coding for the untranslated RNAs. Out of this 5, out of this 7, out of this 5 RNAs, 2 will be play an active role in the X inactivation process, and they are the genes called to be as the exist, XIST, and one more is the TSIX. Exist is going to play a major role because it is going to begin to code the inactive X chromosome. So here exist will be responsible to transcribe the mRNA, which is going to smear all over the X chromosomes starting from the X inactivation center. So here exist causes histone modification as histone H3 methylation and histone H2A ubiquitination, as well as direct modification of DNA, violation of CpG sites. Hence it brings compaction to form the Barrbody. So here exist gene will be responsible to make the many changes of the H3 methylation and histone H2A ubiquitination, and as well as the DNA violation of the CpG sites. So because of this smearing of the whole protein or whatever the exist gene is developing is going to take place. Because of this, the X chromosome, which is taking part in the inactivation or formation of the Barrbody, is going to get highly condensed. Reactivation of the Barrbody is also possible and has been seen in breast cancer patients. A study has revealed the frequency of Barrbodies in breast carcinoma significantly lower compared to the healthy cells. So as we have understood that in females healthy cells, the number of the Barrbodies or the percentage of the Barrbodies will be high. So here compared to this normal females, in the carcinoma patients, that is in the breast cancer patients, the Barrbodies will be significantly lower in number. So this is the normal female cells, which is showing the Barrbody here and also here. Here this is the slide study, the lab slide study, where they have observed the male cells of the nuclear membrane. This is the nuclear membrane. Here also this is the female. Here you can see this is the female. Here you see the Barrbody. Here you see the Barrbody. So here there is no. So out of these pictures you can observe that females showing the Barrbody and the males showing no Barrbody. So thank you for your help tuning to the big book, and spread this particular presentation with your friends and help us to or encourage us to develop more presentations at your service. And thank you guys.